Hi everyone, welcome to the ENCODE Data Collections tutorial. In this tutorial, we will be exploring the different matrices that we have arranged in terms of the different collections. Here I have listed all the types that we will be exploring. The first one is the ENTEx matrix. Next is the reference epigenomes matrix, which is performed on both mouse and humans. Next is the mouse developmental time series matrix. Next is the stem cell developmental matrix. And lastly, it's the ENCORE or the RNA protein interactions matrix. How can you find all of these matrices? In order to reach to the relevant matrix of your interest, you can go to encodeproject.org and click on this button called Data. Upon clicking that button, you will see all the collections listed right here. And as a general rule, we have matrices that are divided into rows and columns. The rows are usually the biosamples and the columns represent the assays. The individual cells represent the number of experiments that are performed on those combinations of biosamples and assay types. Both the rows and the columns are clickable. Once you hover over the columns or the rows of your interest, you can click them and subselect the dataset of your interest. Color-filled cells indicate that we have data available for those combinations. If you find cells that are non-colored or empty, that means no data is available on the portal. We also have search boxes that will allow you to subset your matrices in a quick manner. Let's go over all the different types. The first matrix that we are looking at here is the the ENTEx matrix. It is a part of the GTEx project and it is conducted on 4 human donors. The 4 human donors are listed on the leftmost corner of this page, and here is the expanded view of the same. If you would like to explore what these donors indicate or what their age and different aspects are, you can click on these and reach to the relevant pages. If you are interested in a particular subsample or a biosample type, you can also use the search box, and once you input your search result and hit enter, it will narrow down the query, narrow down the matrix into only the 2 biosamples which are listed here. If you are interested in all the samples that come back on this query, then you can directly go to the list, report, or the download buttons. If you are not interested in all of the samples and want to further subselect using the assay types, you can click on the several assays that we have. In this case, TF-ChIP-seq also has a special meaning where we have grouped all the different the targets that are assayed using the TF-ChIP-seq experiment tag here. If you are interested in all the biosamples across this row, you can hover over here and click on the stomach row, and that will bring you to the list view for all the different types of samples that encompass that biosample. If you would like to know more about the different views on the encodeproject.org, please look up our portal navigations workshop material. We also have the human reference epigenome matrix. This human reference epigenome matrix is comprising of several different tissues, cell lines, primary cells, and in vitro differentiated cells. These are color coded and hence you can distinguish them easily. One of the most important features of this page is the ability for users to subselect their organs of our interest using this clickable human body map diagram. Once you expand this diagram, you can see that all of the organs are listed in this box here, all of the organ systems are listed on the top box here, and all of the tissues are listed on the bottom box here. Even the individual organs are clickable, which means you can either use the text search or the clickable body organs to further explore the data. In this example, selecting the small intestine by clicking on this diagram will narrow down my search results to indicate only the samples that are performed on the small intestine. Next, I'm looking at is the mouse reference epigenome matrix. This mouse reference epigenome matrix is right next to the human epigenome matrix that we looked in the previous page. As of now, we do not have an equivalent body map of the mouse uh, samples, but in the near future, we do hope to implement the same on this page. On a side note, I would like to bring your attention to this button right here. This button indicates that we have more samples that are not easily viewable on this matrix as of now. Clicking on this button will expand the matrix. If you would like to collapse it again, there is a counter button which is called the uncollapse button, and that would just close the matrix. This is the mouse developmental time series matrix. It is an time series matrix where we have several different time points from the embryonic stages of mice. That is exactly, we have 12 different time points starting from 9.5 days to 18 days. We also have two postnatal, that is a 0-day newborn postnatal and an adult of 8 weeks that is assayed as a part of this project. All the biosamples are listed in subtables. That is, we have a separate subtable for each biosample type. This is mainly because the rows in this case are having some extra information, which is the time points. And hence, if you are interested in extracting all the liver samples or all the poly A plus samples on all the different time points, you can still do so. One important feature of this page, though, is the ability to sub-select on time points. If you are just interested in particular time points, for example, 10 days or 10.5 days embryos and an 8-week sample, you can click them individually. Once you have clicked those, it will filter out everything that doesn't belong to those time points. And here is a more collapsed view of the same matrix since those buttons were selected in this case. This is the stem cell developmental matrix. In this matrix, what we are looking at is the different cell types that are derived from the H9 stem cell progenitor cells. The different cells that are derived from this progenitor cell are graphically listed in particular cell types. If you hover over these cell types, you can see the corresponding rows that are highlighted that contain that particular biosample. If you are, for example, not interested in looking at that cell type in this particular matrix, clicking on this cell type will hide that row. Here you can see that since I have clicked on this neural progenitor cell type, that row does not exist anymore, which can be seen right here. If you are interested in bringing it back, just click over that cell type again and you can see them. Or you can use the Show All button. This is the last matrix that we will be covering. This is called the ENCORE matrix or the RNA protein interactions matrix. The ENCORE matrix comprises of around 300 RNA binding proteins, and the project aims at understanding the RNA protein interactions. In this case, we can see that all the target proteins are listed on the left and all the assays are listed on the top. The cells that are primarily used in these assays are K562 and HepG2. We also have a non-biosample containing sample, which is assayed using RNA bind and seq. As you might be aware of, this is an in vitro assay and hence no biosamples are indicated on this column. You can further downsize the matrix, which looks something like this, to a smaller version if you are just interested in specific protein types. For example, if your protein of interest is containing keywords SR, you can start typing them here. And upon typing these, you can see that the matrix is collapsed as compared to this huge matrix right here. And you can further explore by visiting each of these pages that are linked right here. In summary, we have looked at all these 6 different types of matrices, and you are welcome to explore them as per your experiment research projects indicate. We also would like to thank you for your interest, and we would like to encourage you to ask us any questions that you may have on the ENCODE help desk. Thank you very much, and hopefully we can answer all the questions.